So, to understand further the mechanism of transplantation immunology, we will go back to the main important things that we have to understand. So, ano ba to na picture? So, this is a schematic illustration of your HLA complex. So, the human leukocyte antigen complex or the HLA complex are a group of molecules that are present in our cells. So, kasama yan siya doon sa ating major histocompatibility complex. So, as you can see in this slide or in this illustration, nakikita nyo yung locations or structure of your HLA complex and the different structures of the HLA molecule. So, makikita nyo ang different location of genes in the HLA complex, their sizes and distances between genes. Here are just for the purpose of illustration. So makikita nyo that genes that encode several proteins involved in antigen processing, for example, transporter components or components of the proteosomes or HLA-DM, so they belong in the class 2 regions. Ito, class 2 regions. So, in this area naman, the sum of structures of class 1 and class 2 regions are also shown. So, ito ang itsura natin, class 2 MHC and class 1 MHC. So, ganito yung difference nila in illustration. So, yung class 1 MHC, as what we have discussed previously, they are expressed, ito class 1 MHC, expressed on all nucleated cells and platelets. So, they are heterodimers consisting of a polymorphic alpha heavy chain. The heterodimers, so ito, 2, 3, 4. So they are composed of the alpha heavy chain, and that is linked to a smaller non-polymorphic protein, yung beta-2 microglobulin. So yung ating alpha chains, they are encoded by 3 genes. So ano yung 3 genes na nag-encode sa kanya? HLA-A, B, and C. So these HLA, B, and C, they are closely located into one another in one MHC locus na class 1. So makikita nyo yung importance ng alpha 1, alpha 2, alpha 3, and yung alpha 1 and alpha 2 domains will form a cleft or a groove where the peptides bind. Diba? Napag-usapan natin yun last time. So the polymorphic amino acid residues in this illustration line the sides and the base of the peptide binding group. That's why our class 1 alleles can bind different peptides. So si class 1, they can display peptides that are derived from proteins like virus, tumor antigens that are located in the cytoplasm and are usually produced in the cell. So class 1 associated peptides, sino ang type of T cell that will recognize them? Diba? Class 1, CD8 plus T lymphocytes. So the cytoplasmic proteins will be degraded in proteosomes and peptides and are transported into the endoplasmic reticulum where the peptides will bind to newly synthesized class 1 molecule. So wag kakalimutan, so the peptide loaded with the peptide loaded MHC molecules will now associate themselves with this beta-2 microglobulin to form a stable trimer that will be transported to the cell surface. And then there will be presentation of our intracellular peptide to the CD8 plus T cell. So that will now function as CTLs. So that interaction of the T cell receptor and the MHC peptide complex and the CD8 molecule as the co-receptor will um, govern the principle of the MHC class 1 antigen presentations. So again, because of that, our CD8 plus T cells are able to eliminate viruses that can infect nucleated cells and tumors, and they are present in all nucleated cells that express class 1 HLA molecules and that can be detected by CD8 plus T cells. Okay? Inuulit natin to kasi the rejection also is governed by discovering the recognition of transplant antigens from the graft tissues. So, ganun 'di ba si class 1. Ngayon, si class 2 MHC molecule, saan siya coded? Doon tayo sa region called the HLA-D. O, ayan. HLA-D. So, class 2 MHC molecules are encoded in this region with the subregions DP, DQ, and DR. So, and each class 2 MHC molecule, it also exists as a heterodimer, and it is consisting of non-covalently associated alpha chain. Ito. And beta chain. So both of these parts are polymorphic, and the extracellular portions of these alpha and beta chains have two domains, alpha 1, alpha 2, and beta 1, beta 2. So the structure of these class 2 molecules are similar to class 1 because they have peptide binding clefts that face outward. Pero dito, it is the interaction of alpha and beta 1 domains. So that is where the portion of most class 2 alleles differ. So with class 1 molecules, yung polymorphism of class 2 is associated with, just like your class 1 molecules, yung polymorphism of class 2 molecules are associated in their differential binding of the antigenic peptides. So when the class 2 MHC molecules present antigen that are placed into vesicles, they are usually derived extracellularly. And sabi natin, pag merong ni-recognize ang ating mga MHC molecules, ipepresent nila yan sa T-cell. So anong T-cell yung tatanggap ng information from MHC class 2? So yan si CD4 plus T-cell. So the peptides that will result from proteolytic cleavage then associated with class 2 heterodimers in the vesicles and the stable peptide MHC complex will be transported to the cell surface kasi doon magkakaroon ng interaction ang ating MHC and T cell. Okay? So in that interaction between MHC class 2 and the CD4, yung CD4 molecule will act as the co-receptor and the one that will bind is the T cell receptor. Okay? So your CD4 T cells can recognize antigens only in the context of self class 2 molecules. So they are therefore class 2 MHC restricted. And yung ating class 2 MHC molecules are mainly expressed on the cells that present ingested antigens and respond to T cell help. Anong example nun? Macrophage, B cells, and dendritic cells. So also take note that the MHC locus contains genes that encode some complement components and release TNF or tumor necrosis factors and lymphotoxins. So those have the roles in the immune system in recognition of non-self, which we do not want to be exaggerated during transplantation kasi we want our graft to survive in the recipient or the host. Okay. So yung combination ng allele of HLA in each individual, combination ng mga HLA alleles, is the HLA haplotype. So each individual will inherit one set of HLA haplotype from the parent and thus will express two different molecules for every locus. And because of the polymorphism of these HLA genes, we have a varied number of combination of molecules that exist in the population. So each individual will express an MHC profile on his or her cell surface that is different from the haplotypes of most other individuals. So that governs the ability of our cells, of our immune system to recognize hindi talaga to galing sa aking immune or hindi talaga to galing sa aking body. Kasi the HLA profile of these cells that you just gave inside my body is different from myself. That's why, in this case, the immune system is intelligent. Alam niya, hindi niya yun sa kanya. Kasi yun palang pattern of HLA genes and HLA combinations, very varied na. So, there are less likely ability of one ungenetically related individual to another to be similar, except for identical twins. So, kahit parents, mahirap. Kasi nga, combination of one parent to another. Okay? So, ganon ang importance ng ating HLA genes or HLA haplotypes and the MHC profile. So it is believed that the polymorphism evolved so that we can make sure that at least some individuals in a species would be able to display any microbial peptide and will provide protection against any infection. So itong polymorphism, because of the combination of several HLA molecules, means that there are no two individuals other than identical twins that are likely to express the same MHC molecules. And therefore, what does it mean for the transplantation immunology as a whole? It means that grafts exchanged between individuals will be recognized as foreign and will be attacked by the immune systems. That's why important ang roles of MHC molecules because they play a role in regulating the T cell mediated immune response. So, first, different antigenic peptides will bind to different MHC molecules, as we have mentioned. And it follows that an individual will mount an immune response against a protein antigen only if he or she inherits the genes for those MHC molecules that can bind peptides derived from antigen and present it to T cells. So the consequences of inheriting a given MHC gene depend on the nature of the antigen bound by the class 2 molecule. For example, meron kang peptide from ragweed pollen. So the individual who expresses class 2 molecules capable of binding the antigen will be genetically prone to allergic reactions against that pollen. And in contrast, an inherited capacity to bind bacterial peptide will provide resistance to the infection by evoking a protective antibody response. Second, by segregating the cytoplasmic and internalized antigens, the MHC molecules will ensure that there is a correct immune response mounted against those different microbes. So, yung ating cytotoxic T lymphocytes will harbor killing cytoplasmic microbes and yung CD4 naman will mediate antibody and macrophage activation to combat extracellular microbes. And because of this interest in HLA molecules, we realized in the 1960s and 1970s, as mentioned, that there are a number of autoimmune diseases that are associated with inheritance for particular HLA alleles. But that will not be discussed in this video lecture. We have a video lecture separate for that. So, ganon ka-important ang role ng ating MHC and HLA. So, balikan natin paano nga ba nagkakaroon ng antigen presentation. So, the dendritic cells will capture microbial antigen. Yan, capture microbial antigen. And then, it will, it expresses high level of MHC molecules and co-simulators. Now, yung naive T cells natin will recognize MHC-associated peptide antigens displayed on your dendritic cells. And the T cells will now be activated to proliferate. So, na-activate na sila to proliferate. And will differentiate to effector and memory cells which will migrate to sites of infection. So, merong differentiated effector and memory T cells that will enter the circulation and will migrate to the site of antigen. So, yung ating CD4 plus effector T cells of the Th1 subset will recognize antigens of microbes ingested by phagocytes. And yung ating mga CD8 plus cytotoxic T cells will kill infected cells that are harboring microbes in the cytoplasm. Okay? So this is another illustration showing the importance of Th1, Th2, Th17. So these are subsets of helper T cells in response to certain stimuli present at the time of antigen recognition. So the CD4 plus T cells will differentiate into different populations of effector cells that will produce distinct sets of cytokines and perform different functions. So ito yung mga major cytokines produced by subsets of your T cells. Okay? So doon tayo sa mechanism of recognition and rejection of allografts. Kasi sabi natin, the allogenic grafts and the xenografts are the type of transplant that are the most problematic. So yung rejection, this is a process in which T lymphocytes and antibodies produced against graft antigens will react against those tissue grafts and destroy it. So the donor antigens from the graft are recognized by our lymphocytes in the recipient and will produce products that will destroy the graft. So we have several mechanisms. Recognition of graft antigens by T and B lymphocytes, T cell mediated reactions, and antibody mediated reactions. So in recognition of graft alloantigens by our T and B lymphocytes, the major antigenic differences between a donor and a recipient will result in the rejection of transplants that are having a major differences in HLA alleles. So, ang HLA genes natin are highly polymorphic. So, we have several collections or combinations of HLA alleles. So, our allo recognition has two types of pathways. So, meron tayong tinatawag na direct and meron tayong tinatawag na indirect. Pag sinabi nating direct pathway, ito, direct recognition, the T cells of the transplant recipient will recognize allogenic MHC molecules. Ito, on the surface of the APCs in the graft. So, ito yung antigen. Ulit, ito yung allogenic MHC molecules that are presented by the allogenic APCs. So, in this case, the dendritic cells that are carried by the donor organs are the ones that will initiate the antigraft response because they might express high levels of class 1 and class 2 MHC molecules and will have certain co-stimulatory molecules that will allow our recipient T cells to react. So the T cells of the host, once they encounter the donor dendritic cells, will now act, recognize those allogenic molecules and will proliferate and become TH1 subset. Pwede ring TH17. So this direct recognition of the allogenic MHC molecules will now affect the rules of self-MHC restrictions. So yung T cells are recognized are able to recognize foreign peptides displayed by self-MHC molecules. Pero in this case, there is direct recognition of foreign MHC. So at this rate, the T cell will recognize unprocessed allogenic MHC molecules on graft APCs. And because of that, paano ba nun na-detect ng ating T cells? Because there are certain degree of mimicry or resemblance from the foreign MHC and our own MHC. So because of this recognition of allogenic MHC molecules, so that is the cross-reaction of our T cells. And it will create a certain hyperacute rejection response in terms of direct recognition. Tapos pag indirect pathway, ang ibig sabihin lang ng indirect, it means that your T lymphocytes recognize the MHC antigens of the graft donor after they are presented by our own APCs. So dito diretso na yung APC ng allogenic graft will present the allogenic peptide. Pero pag indirect, na-detect ng ating antigen presenting cell, yung allogenic MHC, pinrases pa ng ating APC before pinesent kay T cell. So in this case, this is a processed peptide of the allogenic MHC molecule. So this indirect pathway will generate CD4 plus T cells that enter graft and recognize graft antigens being displayed by the host APCs. And therefore, there is a type of hypersensitivity or inflammatory response that happens here in direct recognition pero medyo delayed kasi pinrases pa. Pero dito sa direct, hyperacute kasi dire-diretso yung pag-recognize. Okay? So that's the difference between direct and indirect allo recognition. So this is another illustration in terms of recognition of alloantigens in organ grafts. So yung direct pathway, the donor class 1 and class 2 MHC antigens on APCs in the graft will be recognized by our CD8 plus T cell. Ito. And will create a certain immune response against that graft. Dito, yung CD4 plus T cells, sa kanya pinresent tong ating recipient APC, yung nakuha niyang MHC peptide from the graft cells. So that is somehow a delayed inflammatory response, pero still an inflammatory or rejection pathway pa din. So it is said that the frequency of T cells that can recognize the foreign antigens in a graft is much higher than the frequency of T cells specific for any microbe. So that said, immune responses to allografts are stronger than responses to pathogens. That's why yung ating immune response can destroy grafts rapidly And that's why, in order for us to make the graft survive, kailangan natin ng immunosuppressive agents, which was recognized by, or discovered by some of the scientists. Okay, so this is just another illustration of the direct and indirect allo recognition. Now, we go to your T cell mediated reactions. So we have our T cells serving a critical role in transplant rejection. So meron tayong tinatawag na acute cellular rejection, pero our T cells can also mediate chronic rejection. So in the T cell mediated reaction, na acute cellular rejection, or also termed as acute T-cell mediated rejection. This is most commonly seen within initial months after transplantation and is shown as clinical and biochemical signs of organ failure. Before, it was thought that the direct killing of graft cells by CD8 plus T cells are the major components of this type of acute cellular rejection. But it was noted also that an important component of this process is the inflammatory response in the graft triggered by the cytokines secreted by the activated CD4 plus T cells. Okay? So that inflammation will result in ano ba yung mga hallmarks? May vascular permeability, tapos accumulation of mononuclear cells, and graft injury that are mediated by activated macrophage. Okay, so yun ang ating acute T-cell mediated reaction. Now, this shows naman that our T-cells can also contribute to the chronic rejection process. So the lymphocytes will react against alloantigens in the organ wall by secreting cytokines. Ito, cytokines. That will induce local inflammation and will stimulate proliferation of vascular and endothelial smooth muscles. Vascular, endothelial, and smooth muscle cells. Okay, so those are your T-cell mediated reactions. So T-cells lang ba yung may kakayahan na mag-mediate ng rejection? Hindi. Pwede rin antibodies. So yung antibodies natin, meron tayong tinatawag na hyperacute, acute antibody, and chronic antibody mediated rejection. So medyo straightforward lang to. So your antibody-mediated reactions will take three forms. So hyperacute rejection, preformed antibodies. So meron ka ng antibody, for example, nakatanggap ka ng kidney ng brother mo. Tapos meron siyang antigens. Ngayon, ikaw as an individual, meron ka ng antibody against those antigens. Yun ang mga antigens. So, even before pa ma-receive mo yung kidney, meron ka ng preformed antibody against those antigens. So, pag binigay sa yung kidney, uy, ready na yung antibody, okay, reject. Hyperacute rejection. So, that's instant. Instant rejection of a graft. So, yun yung hyperacute. Yung acute antibody-mediated is when the anti-donor antibodies are produced after transplantation. So, individual, tapos naka-receive siya ng tissue, tapos nag-produce siya ng antibody against the antigens in the tissue. So, that is acute antibody-mediated rejection. So, in the recipients that are not previously sensitized to transplant antigens, they received exposure to the class 1 and class 2 HLA antigens of the donor graft will trigger antibody production. And the antibodies formed by the recipient will cause injury, pwedeng by to ha, acute. Complement-mediated or complement-dependent cytotoxicity, inflammation, and antibody-dependent cell-mediated cytotoxicity. Okay, so ano ang target ng antibodies in this type of acute rejection? Mostly graft vasculature. Okay, so yun yung acute. Hyperacute preformed antibodies, acute antibodies produced after transplantation. Tapos itong chronic antibody-mediated reaction, ito yung paasa. Akala natin tinanggap ng body yung graft, pero it will develop insidiously without preceding acute rejection and primarily affects vascular components pa rin. So yung antibodies will circulate and will not readily identify the graft, but then eventually there will be some form of vascular lesions that shows graft rejection but are still not well understood. Okay? So same pa rin, the antibodies developed after the graft, pero it took some time for that antibody-mediated response to happen. And mostly, vasculature pa rin yung tina-target. Okay. So those are your T-cell mediated and antibody-mediated reactions. So in addition to the solid organs, there are also variety of organs that are also being transplanted aside yung mga vital organs like liver, kidneys, ganon. And the rejection reaction against some minor solid organs are not as vigorous as what we will expect from the degree of HLA disparity. And the molecular basis of that certain privilege is not well understood. So, pinaka-common na graft rejection can be kidneys, higher than the rejection achieved by lungs and livers. Pero hindi sila yung privilege site talaga. Meron lang minor privilege compared to the kidneys, but not well understood. Okay. So this is examples of different minor types of solid organs. So transplantation and the immune system, ang aim natin, hinayan or i-lesser ang degree of the ability of our body to discriminate between self and non-self because it will destroy grafts. Okay? So antigens of allografts will serve as the principal targets of rejection as proteins are encoded by MHC. So meron tayong tinatawag na transplantation antigen. So yun yung major histocompatibility complex. So if the donor and a recipient of transplant have matching MHC, somehow, the immune system of the recipient is less likely to recognize the transplant as foreign and will not mount a wide or higher degree of immune response against it, thus reducing risk of rejection. So the nomenclature for our transplantation antigens, meron tayong tinatawag na HLA, as we have mentioned earlier. So meron din tayong, tinatawag na SLA, porcine leukocyte antigen. So these are the MHC molecules in pigs, H2 in mouse, and RT1 in rats. So class 1, class 2, intracellular peptides, extracellular peptides. So HLA class 1 yung ABC and class 2 yung HLA-D region. So what are the factors that will favor allograft survival? So blood group compatibility, HLA compatibility, HLA typing, and tissue matching. And the HLA typing will identify the HLA antigens expressed on the surface of leukocytes. So yung recognition of the MHC antigens on another individual cells is one of the strongest immune responses, no, di ba? Is higher in terms of degree than the T cells recognizing pathogens. So, your T cell receptors are very specialized in recognizing MHC molecules. And that selection is the basis for self-MHC restriction of our lymphocytes, which are the fundamental properties of T cells. So, yung histocompatibility antigens, dito nagde-depend ang immune response against transplants. Okay, bisitahin natin ulit ang structure ng ating HLA system. Ito is para yan sa mga mice. So, because of positive selection in the thymus, mature T cells will have some affinity for self-MHC molecules to survive. And those that have high affinity for self-MHC displaying foreign peptides. So yung allogenic MHC molecules that contain peptides derived from allogenic cells may look like self-MHC bound with foreign peptides. Therefore, the recognition of those will result to immunologic cross-reaction as mentioned natin kanina. So, yun yung sa positive selection. That is the one that governs the recognition of our T cells and self-MHC molecules. Ngayon, yung negative selection in the thymus eliminates those T cells that recognize strongly self-MHC. But we have no opportunity to selectively eliminate T cells whose TCR or T cell receptors have high affinity to allogenic MHC molecules because they are never present in the thymus. Kasi nga allogenic sila, galing sila sa labas. So there are clones of T cells specific for different foreign peptides bound to the same self-MHC molecules that may cross-react with one allogenic MHC molecule. Basta yung MHC molecule of that allogenic tissue will resemble certain complex of the self-MHC, pero meron lang siyang foreign peptide na pine-present, which is an allogenic peptide galing sa graft. So, anong nangyayari? Nagkakaroon tayo ng stimulation of an immune response because of cross-reaction. So, we do tissue typing. Meron tayong tinatawag na mixed lymphocyte culture, microcytotoxicity assay, flow cytometry cross-typing, and DNA analysis. Yung mixed lymphocyte culture, we do in vitro model of T cell recognition of alloantigens. So in this model, your T cells from the individual will be cultured with WBCs of another individual, and then the T cell response will be assayed. So yung magnitude of the response is proportional to the extent of the MHC difference between the individuals and is somehow a rough predictor of the outcomes of grafts exchange between these individuals. Although it is in vitro and it does not necessarily 100% parallel the reaction inside, it is still an important prognostic predictor. So that is an indirect representation of alloantigens. So pwede rin tayong magkaroon ng other serologic and molecular tests in determining HLA compatibility. So collectively, these are termed as tissue typing. So the choice na lang depends on what type of organ or tissue in question and the time available for us to tissue types. So we have molecular assays that will be using sequence-specific primers to establish HLA alleles as expressed by the recipient and potential donors. And that is somehow a predictor in characterizing MHC molecules and the ability of the recipient to reject or accept the graft. So remember that the MHC makeup of the donor and recipient is not the 100% sole factor in determining tissue acceptance. Sometimes even when MHC antigens are identical, which will produce a higher possibility of cross-reactivity, it can also cause varying degrees of transplant rejections. And that might be because of minor histocompatibility locus. So, meron tayong tinatawag na non-self molecules that are recognized directly by TCRs, and that yung phenomenon of alloreactivity. And minor histocompatibility antigens are recognized when the peptide fragments of those antigens are presented by some self-MHC molecule. Pero yung sa minor histocompatibility complex, hindi ganun kalaki ang differences but can still lead to rejection of the graft. So sometimes kahit na identical ang HLA matches, there is some degree of immune suppression na kailangan ibigay sa host. Kasi that can be a avenue for rejection by minor histocompatibility complex. So meron din tayong tinatawag na cross-matching of tissues using fluorochrome labeled microbeads that are impregnated with specific HLA proteins. So yung HLA proteins will be associated with a fluorochrome of a different intensity, and yung HLA impregnated beads will be mixed with the recipient's serum. So it will allow clinicians to determine precisely which type of donor-specific anti-HLA antibodies are present in the recipient even before transplantation. So that somehow increases the chance of selecting which host will accept or which type of graft will be accepted by the host. Okay, so these are the different clinical phases of rejection. Ulit, hyperacute, accelerated, acute, and chronic. So here is another presentation of normal allorecognition, indirect and direct recognition. So yung graft acceptance, if the recipient will possess all antigens present in the graft, there will be immune response, and there will be no immune response and no graft rejection, even when the donor and recipient are not syngenic or are isogenic. Ito yung aim natin. Sana may graft acceptance. Pero somehow, karon ang major way natin of allowing graft acceptance is by giving immunosuppressive states. Okay. This just shows the vascular injury that happens during rejection. Di ba yung sabi natin, the target mostly of the acute and chronic rejection are the vasculature. So, paano ba natin kinokontrol? Transplantation immunity is predominantly controlled by cell-mediated immunity. So, puro tayo T-cell. And the humoral antibody can also be produced during allograft rejection. Yung sinabi natin kanina na antibody-mediated rejections. So, the graft will be accepted once we have made the host immunologically tolerant. So, this is another discussion of the chronic rejection. It can occur from months to years, and the underlying mechanisms of rejection can vary depending on the tissue and the size. So, pwedeng sa circulation, lymphatic drainage, expression of MHC antigens, and other factors will determine the rejection rate. So, ang endpoint nito, inflammation, smooth muscle proliferation, fibrosis, and tissue ischemia. So, pag nagkaroon tayo ng fibrosis, meaning yung ating fibroblast nagre-release ng or nag-create ng collagen fibers, rendering that organ unable to function and unable to receive blood supply. So, may tissue ischemia. So, another important scientist, si Gertrude Elion, together with George Hitchings, they discovered important principles of drug treatment for immunosuppression, which gave them the Nobel Prize in Physiology or Medicine in 1988. So these are just some of the drugs that we can use in order to prolong allograft survival. So sige, take a screenshot or a picture tapos tingnan nyo ano ba yung mga mechanisms overview of some of these drugs. So meron tayong tinatawag na privileged sites. So the privileged sites are those areas wherein there are less likely rejection. So fetus is a foreign body, but the placenta somehow acts as an immunologic barrier, although not 100%, especially if there are already IgG molecules that are present that can attack the cells of the fetus. So MHC are present in low density, and the alpha fetoprotein in the blood will help. So another privileged site is the cornea. It somehow survives almost 100% because of lack of vascularity in the eyes or the cornea. So yung transplant noon, hindi nire-reject. Also, the use of hematopoietic stem cell transplant or bone marrow transplant are important for bone marrow failure syndromes or inherited hematopoietic stem cell defects like sickle cell anemia, thalassemia, or immunodeficiency states. So most of the time, we do this transplantation to prevent leukemia. Pero ano yung caveat? Isa sa mga harsh rejections is pag yung mismong donor tissue yung nag-reject sa recipient. So that is a problem unique to hematopoietic stem cell transplantation, yung graft versus host disease and immunodeficiency. So ano ba yung source of stem cells for transplant? Pwedeng peripheral blood stem cells or bone marrow. So ito na yon, graft versus host reaction. So yung graft rejection can be due to the reaction of the host to the grafted tissue. Pero yung graft, pag nag-create siya ng immune response, it can create a response against the antigens of the host. So yung tinatawag natin na graft versus host reaction. So that happens when there are immunologically competent cells or their precursors being transplanted to an immunologically compromised recipient. And those transferred cells will recognize alloantigens in the host and attack host tissues. So that is seen in hematopoietic stem cell transplantation. So yung immunocompetent T cells from the donor will recognize the recipient's HLA antigen. So kung paano nire-reject ng host yung graft, previously, ganoon din ang ginagawa ng graft sa graft versus host reaction when there are immunocompetent T cells present in the hematopoietic stem cells. So one way in trying to minimize this graft versus host disease, yung HSC transplants are done between donor and recipient that are HLA matched using DNA sequence-based methods. So, yun yung idea of the graft versus host reaction. And acute graft versus host reaction can occur within days to weeks after allogenic bone marrow transplantation. So ano ang manifestations? Pwedeng generalized rash, can lead to desquamation and destruction of several gastrointestinal organs. So this is a type 4 hypersensitivity reaction. So cell-mediated T cells antibodies plays no role in the graft versus host disease. Okay. So the recipient should not reject the graft, and we hope that the graft will not attack the recipient. Okay? So fetus, yung sabi natin, being protected by the placental barrier para hindi siya i-attack ng system ng mother. So as a recall, these are the clinical phases of rejection. Again, rejection response against solid organ transplant is initiated mainly by host T cells that recognize foreign HLA antigens on the graft, either directly, yung direct talaga yung APCs ng graft, nagpe-present ng peptide, or indirectly wherein APCs ng host, yung kumuha ng peptide, ipe-present kay T cell. So hyperacute rejection, pre-existing preformed antibodies. Acute cellular rejection, antibodies that are created after the tissue transplantation. And chronic rejection, mostly mixed humoral and cellular mechanism. So hyperacute rejection, nagkakaroon tayo ng thrombosis, ischemic damage, and rapid graft failure, minutes to hours. Acute cellular rejection, it is remediated by inflammatory reactions, complement cytotoxicities, and destroying vasculature. Tapos, yun namang ating chronic rejection is a combination na of the different inflammatory processes. Mainly, vasculature pa rin yung ina-attack and nagkakaroon ng parenchymal fibrosis. Okay. So, treatment of graft rejection relies on immunosuppressive drugs, and the transplantation of hematopoietic stem cells naman can make or can present rejection by graft versus host disease in immunodeficiency. Okay? So that is the transplantation immunology. I hope you learned a lot, and please don't forget to study your book, and God